It's my pleasure to introduce Sarah Haines, who's a professor in the Civil and Mineral Engineering Department at U of T. And so Sarah comes to us from Columbus, Ohio, where in the past, before she came here, she did her undergrad, master's, and PhD at The Ohio State University. BS in environmental engineering and MS and PhD in environmental science. And her research focuses on building science and indoor air quality, so a lot of overlap with what we do here. And so she aims to combine building science, engineering, and microbiology to understand the impact the built environment has on human health. So research topics in her group include housing, mold, and moisture, and using cutting-edge microbiology techniques such as next-generation sequencing, metatranscriptomics, and bioinformatics to look at the indoor microbiome and how it impacts health and building integrity. So with that, let's welcome Sarah.
Great, thank you. Thank you, Arthur. Okay, so I believe I'm screen sharing. So unfortunately, I don't think I can still see you guys once I start screen sharing, but feel free to.
We're still here, don't worry.
Okay, yeah, and just unmute if anything comes up or you have any questions. So yeah, so thanks so much. I'm really excited to talk to you all today. Thank you for inviting me to the SOCAAR Seminar. And today I'll be giving kind of a background of my research and where things are headed in the future. So with that, let's start off. So we spend about 90% of our time indoors, and I'm sure you've all heard this before, especially during COVID when we've all been stuck inside, or during these cold months where we're just spending so much time indoors thinking about all of the different things that we could potentially be exposed to. And so some of our indoor exposures, they can occur from a variety of different sources. So we have chemical and microbial sources. So things in our indoor environment can come from outdoor air or from the people that live in our environments, or animals, pets, plants, as well as things from water. So there's lots of different pieces and parts of our indoor environment. And so with my research, I'm always thinking about, what are these exposures? What are the chemical and microbial exposures that could be impacting our health on a daily basis? And one of the things that I really focus on, or have been focusing on in my research, is dust. And so carpet and dust. And so carpet and dust are actually a source of both microbial and chemical exposure. So within dust, there's lots of different microorganisms, and then there's also different chemicals. And one of our main exposures to these in the indoor environment is due to resuspension. So when we're in an indoor environment and you have dust all over the floor, and maybe even when you're sweeping or vacuuming or you're just walking around, that dust becomes resuspended, and it can come in contact with our upper respiratory system as well. And so thinking about how, this resuspension of dust and all of these different microbes or chemicals in our dust be coming in contact with us and impacting our health. And what I've been focusing on is moisture. And so we know that, as I said, there are microbes in our dust, but not only are they just in our dust and in our environments, they can actually grow at elevated relative humidity conditions. And so in this figure that I have on the left, we can see that as you increase equilibrium relative humidity across the x-axis, you're actually increasing the spore equivalents per milligram dust, or that total quantity of growth. So as you increase moisture, we often see an increase in growth. And in the picture on the right that I provided, that's actually a piece of carpet that I incubated. I sprinkled dust on it and incubated it at 100% equilibrium relative humidity for 2 weeks. So that's lots of moisture. You can actually see the mold growing on the carpet. So hopefully you don't have this in your home, but this can happen, right? If we have all this moisture, microbes can be growing in our dust and on our building materials. And not only are the microbes themselves growing, but they can also release microbial volatile organic compounds. And so these are those MVOCs, which are those moldy and musty odors that you often smell when mold is growing. So I like to think about your moldy sponge, and when, maybe you're cleaning your dishes and you can smell, something doesn't smell right. And that's those MVOCs, those microbes that are growing in your sponge, if you start to smell MVOCs coming off of your sponge, throw it away, right? There's probably something growing in that. Time to get a new sponge. But these microbes actually release these compounds into our environment, and they can potentially impact people get headaches or impact your upper respiratory system, your eyes, nose, and throat. So there are some impacts from microbial volatiles as well. And exposure to mold and dampness in housing is actually associated with asthma and upper respiratory disease. So when we're getting this increased moisture and dampness and mold in our houses, we can see an increase in prevalence of asthma and upper respiratory disease. So with my research, I really want to understand microbial growth indoors, and as it is critical to building science overall. So hopefully no one has a home that is completely covered in mold like this beautiful artwork that we see here, but microbes can be growing in our homes. Moisture can be impacting the release of those MVOCs, VOCs from the materials themselves, as well as just the microorganisms that are growing in our indoor environments. And so my research really focuses on this relationship between moisture, microbes, chemicals, indoors and their impact to human health. So kind of thinking about all of these different aspects and relating it back to our exposures in our indoor environments. So to start off, I'm going to talk a little bit about the influence of moisture on dust and carpets. And so our home environment, it does not stay at a constant relative humidity throughout the day, right? It's not going to just stay at 70%, 60% relative humidity the entire time. It varies with temperature and other factors depending on the home or indoor environment itself. And actually, we've seen in previous work that the room air relative humidity can actually be lower than the relative humidity of the carpet. And so if you're thinking back to that image I showed you earlier of that mold growth on the carpet, if we're clocking in the room air relative humidity as maybe 65%, but then down in the carpet, we're actually seeing a higher relative humidity. Maybe we're seeing more growth occurring down at that carpet as it's hygroscopic and keeping that moisture in. And so what I wanted to do was determine if we could model fungal growth in carpet dust using this previous time of wetness framework. And so the time of wetness framework was developed for growth in drywall. And what they determined was that after relative humidity was at or above an 80% threshold, so once you reach 80%, that's when you start to see fungal growth on drywall. And so for this research, we wanted to determine, can we use this framework to model fungal growth in carpet? As I was mentioning, the carpet and dust, that's where we see lots of our exposure to these microorganisms. So to do this, I actually traveled, as Arthur was saying, I'm from Ohio. I traveled all across the state of Ohio. I would go into people's homes and rip out their carpet. So you can actually see the images there where I took carpet. Don't worry, they were getting rid of it. I didn't just go and take a giant chunk of carpet and leave. They were getting rid of it anyway. And as well as I vacuumed the home and collected all the vacuum bag dust. So we had carpet, as well as vacuum bag dust. I would take the dust from the same home and embed it into the carpet, and then we would incubate the carpets at varying relative humidity conditions and in these little incubation jars. And so we would vary the relative humidity conditions between either 50% or 85 or 50% and 100% relative humidity for about 14 days, and this was to create those diurnal variations in relative humidity that you see in your home. So as I mentioned, your home isn't going to stay at a constant relative humidity throughout the day. So by varying these conditions, we could actually simulate what potentially would be experienced in a home with some elevated moisture. And so after 14 days, we would then vacuum up the dust. I would run DNA extraction, so extract any of the DNA from the dust. We ran quantitative polymerase chain reaction, qPCR, which some of you may be more familiar with now with COVID, getting your PCR tests. So basically, same thing, but instead of testing for COVID, we were testing for bacteria and fungus DNA. And then, so once we had the quantity of growth from qPCR, we would then determine the actual species composition, and that was done through Illumina-MiSeq DNA sequencing. So then we could actually determine, what bacterial or fungal species are growing more prominently at those different relative humidity levels. So results from this work, we actually found that fungal growth in dust in carpet follows this 2 activation regime model. And so the 2 activation model actually accounts for the lag time that we see in fungal growth. And so if you're looking at this figure, we see on the x-axis is the time of wetness. So that's that fraction of time that a sample was above the 80% critical growth threshold, or the 80% relative humidity level. And then the y-axis is our relative growth rate. And so there is a bit of a lag in between 0 and the 0.25 time, and that's likely because there's the fungal growth that's there, we're accounting for that lag time. So it takes them a little bit of time to get used to, oh, there's some moisture, and they start to grow, and then it kind of evens out, and then we see some exponential growth as the relative humidity increases for a longer period of time. And so we can actually model fungal growth in carpet dust. Additionally, we wanted to look at all the different fungal species that we were seeing within those dust samples that we collected. And so what we expected to see is that at a higher relative humidity condition, we're seeing all of these really common fungal communities, but that's actually not really what we were seeing. So each of the different colors are a different home. So that's a different home that I collected dust and carpet from. And then the similar, the different shade of color is the relative humidity condition. So a darker color would be a higher relative humidity condition, so like 100% for 24 hours. And what we're looking at this chart are similar fungal communities clustering together. And so what we found was that actual fungal communities were more similar to each other in one home than they were to anything else. So the fungal communities, say, in my home are more similar to each other than they are in a different home. However, when you think about the fungal communities within one home, we actually see that moisture does influence the microbial communities that are in that one home itself. But then we wanted to know, okay, well, what does this growth look like? So we know it's occurring. We know that dust, the fungus is growing in the carpet dust. What is actually happening? And so we had these beautiful SEM images done where you can actually see as you're increasing the relative humidity, we have nylon fibers, we have dust, and as you increase, you can actually start to see the hyphae, the spore chains on the nylon fibers. Interestingly, you need the dust for growth, is what we found. So the dust actually contributes to the overall growth. If you don't have any dust on your carpet, likely not going to get any microbial growth, as that was very key to this growth occurring. Potentially, the fungus is using the dust as a carbon source or for other sources of growth. Additionally, we wanted to also look at allergen concentration. So not only fungal growth and those different microbes, but also what kind of allergens could be coming off of those microbial communities at these different moisture conditions. And what we found was that as you increase the relative humidity, it is associated with increasing allergen concentrations, specifically this Alt-A1 protein, which has potential negative health implications. Okay, so now moving from kind of our indoor environment to the ultimate indoor environment, which is the International Space Station. So I was actually lucky enough to work with dust collected on the International Space Station. It wasn't dust, it wasn't moon dust, it wasn't outside in space, but it was up in space. It was dust on the International Space Station. So previously, there have been moisture issues on spacecraft. So we saw that on the Space Shuttle Mir, which was pre-International Space Station, there was a series of power outages that caused a free-floating condensate to occur. So there's lots of increased humidity and temperature on that spacecraft, and a water ball was just kind of floating around, and they actually found this amoeba growing in it. And then additionally, on the International Space Station itself, the astronauts were working out, they're running, and they're sweating, and they would hang their sweaty towels onto those racks and that were up against fabric panels, and we saw mold growing. So what we wanted to look at, is there's differences in growth, but also with particles. So if we're thinking about particles on the International Space Station, you're not actually no particles are going to settle out. So when I was talking about resuspension of dust in our home environment, that isn't really happening on the International Space Station because there's just the dust isn't going to settle, there's zero gravity. And so on Earth, we see particle sizes influence the bacterial and fungal genomes. And so at different particle sizes, we actually see differences in our bacterial and fungal genomes depending on the actual size. But when I analyzed the fungal concentrations and bacterial concentrations of dust particles taken from the International Space Station, we see something a little different. And so these are actually higher particle sizes than you would likely see resuspended on Earth. We often see particles above 100 microns settling out, but these particles, you know, they are larger. We're seeing all these different fungal concentrations and bacterial concentrations on these different sized particles that could potentially be coming in contact with upper respiratory systems of the astronauts that are on board these different spacecraft. So I really enjoyed this work. I'm interested in seeing how, you know, further International Space Station work. I know it is being decommissioned. So as we start moving to, you know, different environments out in indoor environments out in space, things like this, you know, we need to be thinking about how we could be impacted, because you're likely going to be inside for most of the time if you're, say, living on the moon, right? Okay, so to continue on to talk more about the chemical and microbial interactions indoors. And so dust actually contains lots of different things. As I was mentioning, it contains microbes, it contains chemicals, and one of the other pieces is it contains phthalate esters. So if you have heard of phthalates, they're common plasticizers and endocrine disruptors found in a variety of products. They're really found in so many different things, and they're commonly found in our dust. They're everywhere in your home. Your home probably contains lots of phthalates. And so what we wanted to know is, can phthalates in dust be degraded due to microbial or chemical processes? So can these phthalates, could this be helpful? Could we be degrading these harmful phthalates that are in our dust through either, you know, microbial means or chemical means? And so what we did was actually take dust, and it was a whole incubation experiment again, as I had explained earlier. And similarly, we would incubate the dust at different relative humidity conditions and then analyze, are phthalates in the dust being degraded due to the microbial growth or due to other chemical processes that are occurring, so potentially not microorganism growth. So we did find in this study that the DDEHP, which is a type of phthalate, was degraded due to both abiotic and biotic processes. And what was interesting, though, is that the degradation of this DDEHP would actually lead to byproducts such as MEHP and phthalic acid, which may be more irritating and harmful than the DEHP itself. So potentially, yes, we could be degrading these phthalates in our indoor environments, but potentially the degradation byproducts could be more harmful to health. So again, it's truly important to analyze these chemical and microbial processes in our dust and in our carpet. And so next, what I wanted to look at was, how does material type actually impact the growth as well as the release of MVOCs? So taking another step further into this relationship between moisture, microbial growth, as well as release of microbial volatiles or volatile organic compounds themselves. So what are the emissions from the materials, things like carpet, drywall, the dust itself, as well as emissions from the microorganisms themselves? And so for this experiment, we actually collected dust from different parts of the United States, so different climates, Ohio, Florida, and California. I would sieve the dust to obtain those fine particles, embed the dust into carpet. And again, we did our incubation experiments where we were incubating carpet and dust, as well as this time drywall, incubated in triplicate at 50, 65, 70, 75, 80, 85, and 95% relative humidity. And so we wanted to incubate them at these different levels of relative humidity to determine what kind of emissions would be coming off either the carpet or the drywall itself, as well as from the actual microorganisms at these different levels of relative humidity. And so to get a large range of potentially what could be the different ranges of relative humidity in a home environment. And so after these samples were incubated for about 4 weeks, we would sniff them using the proton transfer time of flight mass spectrometer, so the PTR-TOF-MS. And so we were actively measuring emissions in real time. So air would be flushed through and then sucked back out, and the emissions were measured based on their mass ratio. And so we could actually determine, what emissions are coming off of these samples in real time. Again, I would collect the dust or take a swab if it was a drywall sample, run DNA extractions, qPCR sequencing, so we could determine what the quantity of growth was that was was occurring, as well as the species type. And so initial results from this, we actually found that fungal growth in dust actually occurs at a lower relative humidity than growth in drywall. And so you may be thinking, you know, these figures look a little similar to maybe some that I was showing earlier, which is very true. But in this analysis, we were comparing drywall and carpet. And so we can actually see that the growth in drywall, the growth in drywall didn't occur until you were at a very elevated relative humidity condition as compared to the growth in carpet. And so again, it's important that we're thinking about the growth in dust and carpet, as this is where we could be seeing these exposures. If you're kicking up this dust, you're coming in contact with it, these are where we could be seeing these potential health exposures. Within the material type and sample location, we actually saw something a little bigger than that previous study where I was showing you that was all from Ohio, all those samples from the same state. In this analysis, we were actually seeing, you know, similar species composition within the different locations themselves. So very similarly, things were separating based on geographic location. And that was actually influencing the fungal and bacterial communities within a material sample type. The sample type itself also influenced the fungal and bacterial communities as well. So if it was dust or carpet with dust or just drywall alone, there are different communities growing within those different sample types. But again, how do moisture availability and substrate then impact this MVOC and VOC production? So we know that these microbes are growing on these different materials, but how is the moisture actually impacting potentially the release of MVOCs from the microorganisms or the VOCs that are produced from the materials themselves? And so interestingly, what we found was that moisture and the presence of dust actually influence the concentration and the emission type. So initially, in the top figure that you're looking at here, this is dust and carpet samples. So this was the dust and carpet that was incubated at those different equilibrium relative humidity conditions. And so initially, we had assumed that as you increase moisture condition, perhaps you would also increase the actual total emissions that are coming off of the material and the carpet itself. So things would be, you know, VOCs would be being released from the material, from the carpet, as well as MVOCs from the microorganisms that could be growing at those elevated humidity conditions. That is not especially what we are seeing here. So we actually are likely seeing a dampening effect from the dust on the carpet sample. So when we included dust within the carpet, the actual VOCs that were released from the carpet itself, which you can see on the bottom figure, which is just carpet, no dust, you see a lot of different emissions coming off the carpet at 95%. But when you add the dust, potentially, you know, those emissions aren't being released anymore, or perhaps the microorganisms themselves are somehow using the VOCs or these emissions for growth or for some other processes. And so we actually see that when you add dust at these different humidity conditions, there are different types of VOCs that are being released, as well as when you don't have dust, you start to see, again, just different types of volatiles that are being released. And so we did see some MVOCs, some common MVOCs that are known in indoor environments, such as monoterpenes, as well as dimethyl sulfide, among others. And these are commonly associated with microbial growth in indoor environments. And so we did see these. They were associated with those carpet and dust samples at 95% relative humidity, which is where we would always expect to see growth occurring. Additionally, we saw that dynamic relative humidity conditions actually impact the VOC emission profile from carpet. So this is a figure actually just showing carpet alone. So this was without dust. But over time, we were varying the relative humidity between either 50% or 85%. And you can see that there is a spike, right? So as you switch from 50% to 85%, there's a spike in emissions, and then it would go back down. This is not what we saw. The emissions that we were seeing here are not the same types of emissions that we were seeing if we just left everything consistent at either 50 or 85%. actually seeing lots of nitrogen containing compounds when we varied those conditions. And so this may be more similar with what you may see in your home. As again, you're not keeping everything at a constant relative humidity. So if the moisture is being varied, potentially you could have more nitrogen related compounds being released from these materials. Key takeaways from this research that I've gone through is that microbial growth can be measured in carpet dust using the 2-activation regime model. Dust in carpet requires less moisture for growth than growth on drywall. And the material type likely releases a large percentage of VOCs, and moisture influences both the concentration and the diversity of compounds that are released from these different materials. So now I'm going to go into a little bit more of my current work that I've been working on since I began my time here at U of T. So I did just start my faculty position in July, but we have some things up and running already that I'm excited to share with you. So I've been continuing to work with Dr. Pavel Mishal at UT Austin, as well as Dr. Ashley Bope at The Ohio State University, to determine how moisture impacts gene expression in dust as well, and how that relates to the release of microbial volatiles. So with this work, we want to kind of dive in further with what I was previously talking about and look at how not only what microbial volatiles are being released from these dust and carpet or just dust alone, but what how does gene expression play into this release of compounds? So what genes are being expressed? What are the microbes actually doing when they're releasing these compounds? And we currently just don't have much of that information. Some previous work in this GIF that I have going here was seeing how increase of moisture can actually increase the gene expression. So as at 50%, there's some, something going on, and then at 100%, all of those different pathways start to light up. So as you increase moisture, we often see an increase in gene expression. Now we want to analyze how these gene expression could be relating to, or how it's working in relation to the release of microbial volatiles. And so some initial results, we are actually seeing higher expression of genes at 50 and 95%, which is interesting when compared to moisture at 85%. And so this is likely due to the fact that higher gene expression is occurring at that 50% for primary processes that are essential for cell maintenance. So perhaps that's not where we're seeing any release of MVOCs, but that the 95%, there is an upregulation of secondary metabolic processes, which is consistent with that previous study and that GIF that I showed you. And so potentially that is where we could be seeing these release of microbial volatiles. Perhaps those genes, when those are being expressed, that's when those MVOCs are being released. Additionally, I was part of a study looking at bulk floor dust in relation to monitoring viral disease outbreaks in buildings. And so in this study, we actually collected dust from rooms of positive individuals, those who had tested positive for COVID-19, and we were able to detect SARS-CoV-2 RNA in the dust as well as on other samples. But what we were actually finding is that in the dust, you are seeing a high amount of RNA. This does not mean that it is infectious. This is just the concentration of RNA, of SARS-CoV-2 RNA that was in that room or in that environment. And so we can actually potentially use dust as a matrix for monitoring viral disease outbreak. And so if you've been hearing about the wastewater monitoring that some fabulous researchers here at U of T have been working on, we are hoping to utilize vacuum dust and dust in the air as a way to monitor disease outbreaks. And so I have my master's student, Zoe Hoskin, working on this in collaboration with Dr. Jeffrey Siegel here at University of Toronto. And we are estimating airborne concentrations of SARS-CoV-2 using these filter forensics techniques. So we have air cleaners that we have been deploying in homes of those who have tested positive for COVID-19. We deploy them in indoor environments. We leave them for about 2 weeks, and dust is collected onto a filter. We vacuum off the dust on the filter, and we have flow measurements, so we know how much flow, the rate of flow pre-deploying the filter as well as post-deployment of the filter, so we can determine the flow rate over time. And then once we collect that dust, we actually run RNA extractions and qPCR. So again, very similar. If you ever had a PCR test done, they were also extracting RNA from your nose swab. We are extracting it from these dust samples. We're actually able to determine the quantity of SARS-CoV-2 RNA in those dust samples on the filter. And so again, this is not infectivity, this is just the concentration of RNA copies that were potentially in the air at the time when this person was had come in, when the person had COVID-19. Sorry. So some initial results that I'm excited to share with you is that we are seeing the presence of airborne SARS-CoV-2 and RNA changing throughout the home of a positive individual. And so in this home, we actually had an air cleaner in a bedroom. So the person was theoretically isolating in their bedroom for the period of time where they had tested positive. And we had an air cleaner right outside of their bedroom. And we also had an air cleaner down in the common room. So they were not leaving their bedroom, but we did see the presence of SARS-CoV-2 RNA, average copies about 1.6 copies per meters cubed of air in the room where they were, but we were also seeing RNA copies right outside of their room, as well as in the common area. So it is potentially, it is an airborne virus, and that RNA was detected throughout the home. Additionally, we were detecting for humidifier usage in the home, so moisture. So in certain homes where we deployed these air cleaners, there was no humidifier, and in certain homes, there was a humidifier somewhere in the home, but it wasn't in the same room as our air cleaners. So in this chart here, we can actually see that the flow rate of our air cleaner decreased almost exponentially as in the rooms of those that had humidifiers in the actual room. So we see pre-deployment and post-deployment. So if there was no humidifier, the flow rate, it decreased a little. That's expected as you're loading dust. In a home where there was a humidifier in the home, but maybe it wasn't in the same room, we also saw a decrease. But then in homes where you had a humidifier in the room, you had the air cleaner, the air cleaners were returned to us with a thin layer of white dust, and the flow rate was dropped very, very heavily, as you can see, from about 12 to maybe 1 meters cubed per hour. So that was really interesting. We're going to be looking more into humidifier dust and how that can be impacting the usage of air cleaners in homes. Additionally, so some work that I'm hoping to get started here as I begin my time at U of T is looking at mold and dampness in low socioeconomic communities as well as Indigenous communities. So mold and dampness does disproportionately impact these communities, and I've actually been working with someone directly who works with those in Indigenous communities. I wanted to share the pictures with you all, but I don't have access to them, but there are pictures of just mold all throughout these homes. It's, absolutely horrible. And so my hope is to go into the homes and be working directly with the communities to determine new ways that we can, better ventilate homes or decrease this harmful mold growth that is occurring. And so potential ways that future technological advances, we may be able to use chemical detection of microbial volatiles indoors. So I was previously on a project to detect formaldehyde indoors using a smartphone app with a color changing badge. So the color change would actually occur depending on the formaldehyde concentration. So if they're, depending on how much formaldehyde was in a home, potentially things like this, using smartphones, we could be using that to detect mold in homes that we can't visually see or smell. Okay, so with that, I would like to just thank all of my many collaborators, and I will take any questions and happy to talk with you with how much time we have left. So thanks so much, everyone.
Well, thank you for a wonderful presentation. It's both interesting and kind of scary. So But we do have lots of time for questions. I see some in the chat, but we could start with those, or I see a hand up. Let's start with a couple online. We can go to the chat. So let's see who we got here. The hands up. Taylor.
Hi, thanks. I have so many questions, but I'll try to stick to one because I don't want to monopolize your time. I was really interested when you talked about the differences in particle deposition when you have the microgravity on the ISS.
Yeah.
And I was surprised that you mentioned that you wouldn't normally find deposition, because I expected intuitively that rather than large particles depositing quickly due to gravity, that large particles would still deposit quickly, just because with their regular Brownian motion in the air, they would have more momentum when they collide with the walls eventually. So did you ever explore any of like maybe the mechanistic reasons why you were seeing the distributions that you did in those environments?
Yeah, that's a great question. So you're saying on the International Space Station in zero gravity, like could there still be deposition with the large particles? Is that yeah, so I think that's a great question, and I would love to explore that further. So we didn't do any it wasn't a health related study, so we were just looking at, the particle sizes. It was basically we were sent this dust that someone else had been using for chemical analysis. We kind of got our hands on it to look at the fungal and bacterial communities. But I would agree, it would be really fascinating to actually determine, are these particles, because they are larger particles, do they end up getting, some kind of deposition filtered out of the air on the ISS more quickly than the smaller particles? I'm actually not sure. I know I was going to mention, so they do have on the International Space Station, they have an airflow kind of system. So they have I believe it's a HEPA filter. And so air is being pulled through. But as you're floating around, it's still, there's particles that the astronauts complain that things get stuck in their eyes. Lots of them end up with different kind of skin rashes when they come back from the International Space Station. We're not sure, what that's coming from, but many of them have skin rashes. But yeah, the particles kind of flow through the air and they get trapped on the HEPA filter. You hope, right? So I think that would be really interesting if you could actually, in real time, maybe be vacuuming off the HEPA filters and be seeing what kind of particles are on those filters. That would be great. Yeah.
Thanks.
Yeah.
Very interesting.
I'm sorry to interrupt for a second, Sarah. There's someone in the waiting room that had been kicked off and was trying to get in. I wonder if you have the option, because I don't.
No, I just got in now. Just came in.
Okay, great. Thanks.
Great seminar, Sarah.
Thanks, Jeff.
Unfortunately.
I see some questions. Let's go to the chat. So there's a few there. Actually, quite a few.
Okay.
Yes, there's lots. So Lauren, you had a couple questions. Why don't you start with that?
Okay, so there's some minor construction in my house, so I hope that for the time that I'm speaking, you don't hear it. But I know you mentioned that you had found some nylon fibers that were actually promoting the microbial growth, or that microbial growth needs dust to survive. So
yeah.
With COVID, we've heard plastic surfaces can actually promote just longer survival of COVID. And I was wondering if that's something you kind of observe with your research. So are plastic fibers more conducive to microbial growth than non-plastic materials?
Oh, yeah, that's an interesting question. Yeah, so what we were looking at were different types of carpet fibers. And so often carpets are made of, it's nylon or what is it? Not polystyrene. It's one of the polyethylene, or I'll look it up for you. But they're made of those different fibers, and that's what we commonly see. So I wasn't checking plastics, but I think that's a good point. Like we do see often potentially like biofilms on certain plastics as well. So it would be interesting to see if you were using other types of materials in your home, could that be promoting? Ideally, so what we were finding with the carpet and the dust was that most often, no matter what, as long as you had the dust, that is when you were seeing growth. We did see higher concentrations of growth in, I believe, wool fibers, which are, more naturally occurring fibers. So we were seeing lots of growth in those, but there was similar growth within nylon fibers and olefin, I believe, as well. And so, yeah, I haven't looked too much in plastic, but that would be interesting to see, if dust on plastic could be influencing growth. Yeah.
Thanks so much. And then also one last question, because I know you said that the communities growing on the particles varied with their size distribution. I was wondering what the size detection limit of the particles were.
For the space station one, or for?
I think it was in the home. It was on slide 24, I think.
Oh, yeah, here we can go back.
Curious. Yeah, I'm just looking
Yeah.
at like plastics in the air, so I was really
Oh,
excited.
okay. Yeah, maybe we can chat. Okay, slide 24. Yeah. So this was basically this was also from the International Space Station. So these were the particle sizes that we were given. So I would love to have a giant vacuum bag of dust from the ISS and actually, sieve it out and get those fine particles. But for this study specifically, unfortunately, these were the size particles that were sent to us that we were able to analyze. So we only had, between 53 to 500 micrometers. Yeah.
Okay, thanks so much.
Yeah, of course. Thanks for your questions.
I've got a follow-up question, and then we'll see if there's others online. So for the in-home, you showed us some micrographs of particles, but I didn't quite catch the scale. Are they, when you're talking about these particles, are they 100 micron particles, or are you seeing growth at the particle sizes that we more often associate with airborne? So,
these were sieved. These were sieved to fine particulates. So this is what we would often see in I sieved it all down to very fine particle sizes. So that is more what you would be likely seeing with deposition rates or resuspension. So they're smaller particles. I can check exactly what the size fraction was. I'm trying to remember what sieve I used.
Yeah, sometimes it's hard to, well, actually, we can get down to sort of 50 micron, but sieving down to micron is.
It was hard. It was, it's gross to sieve the dust. So I would just have vacuum bags. So who knows, there's hair, there's dog hair, pieces of toenail clippings. And so I try to sieve all of that out as well.
Great.
Yeah.
Let's see, James, you had a question.
Yeah, I just thanks for a really great presentation, first of all. But I found the variable humidity conditions finding really interesting. And like when I think intuitively about like seasonal variation in humidity in homes, there might be more variability in like the seasonal periods when you would also see a lot of pollen. And traditionally, seasonal allergies are so frequently attributed to pollen. But I'm just wondering, what the cross feature of the MVOC sort of production might be in those variable humidity conditions. So really interesting. I wonder if there's any studies that look at both.
Yeah, now I'm trying to remember if they did moisture, but there was someone at the previous lab that I was working on, they were specifically looking at seasonal allergies in relation to dust and how the presence, depending on the season, how much growth was occurring. I'm not sure if they were looking at relative humidity in the homes though, because that would be fascinating to see, over time. Because I know, I just moved here, but August was very humid in my apartment. So thinking about changes throughout the year, and now it's very dry. So absolutely, how does that relate to allergies as well?
Very cool. Thanks.
Yeah.
Sarah, can you see the chat?
Yes.
Why don't you pick some questions? There's
Okay.
quite a number of them.
Okay.
So pick some that are easy.
Okay, Michael asked, if dust is a source of growth, is that mean that we have a lot of microbial growth on HVAC filters? If so, they'll be very dangerous. Yeah, that's a good question. So I would say as long as you're not, I should have had a disclaimer at the end, as long as your humidity in your home is not constantly, you're not getting above this 80% threshold what we're seeing, there shouldn't be that much growth. So hopefully on your HVAC filters, it's not super humid in there, so you're not going to be getting growth. Hopefully they are just collecting any microbes that potentially could be maybe growing in your carpet or other areas of your home. So I know there is lots of research on the coils of air conditioning units. So you do see that often if there's dust, and I'm not sure what the coils are made out of, but they do see microbial growth on the coils in air conditioning units. And that's why sometimes when you run your AC, and if you have one of those little units and it kind of smells moldy, musty, that could be from that. Let's see. Oh, okay. So for homes, we have for homes with carpet, could humidifier be used? So I'll answer the first part. Would vacuuming help reduce microbial growth? So I have, I'm not going to say that you shouldn't use a humidifier, because I know often, it does get really dry in the winter. So I would say if you need to have a humidifier in your home, use a more of a hot steam humidifier is what we were finding, especially when I was talking about the COVID filters where they were covered with that white dust. That's often from those cold mist humidifiers that are you're just putting tap water in, and it's just, coming from the water itself as it's misting. So I would say hot mist humidifier or using distilled water in a humidifier. Vacuuming to help reduce microbial growth. Yes, absolutely. In the sense of keeping any dust out of your carpet. I would highly suggest vacuuming your carpet if you have one weekly. I always did. I don't have carpet in my home anymore after doing this research for so long. I specifically don't have carpet. But if you do, and I did for a long time, definitely vacuum. Okay, let's see what other questions we have. Arthur, did you want to ask your question?
Yeah, sure. I'm still recovering from what you said about vacuuming. And so, yeah, nice talk. It's a lot of interesting things. My question was regarding the human occupancy inside a home. Whether for your projects where you collected dust from homes, do you have separate occupied homes, dust from occupied homes and dust from unoccupied homes?
Yeah, that's a good question. No, so we only had dust from occupied homes that were currently being lived in. I'd have to go back and check. I know there was a couple homes where there was a period of time where they were renovating, and so no one was living there. So it could be interesting to compare. But I think that's a good point. It can depend on how many people are living there, presence of pets, what microorganisms. We were hopeful to see some kind of, you have a dog, and these homes all had dogs, so maybe we're seeing these similar fungal communities, and it wasn't like that. It was just very different. The communities were just so different depending on maybe where their home was or who was living there. But the microbial communities in your house change. So if I moved out and someone else moved in, they would start changing immediately when as you occupy the space.
Yeah, because I've heard from, I think it was Charlie Weschler who said that like house dust has a ton of skin flakes from humans in them.
Yes.
And it's in your clothing, it's everywhere.
Yes. Most of dust is skin.
Yeah, so I was wondering if like the microbes were sort of eating off of that, and that could be an interesting sort of symbiotic relationship between us and the microbes inside a home.
Yeah, definitely. I think that's one of my hypotheses as what's happening. They're eating, they're using the skin flakes as a carbon source, but it's hard to say what exactly they're using in the dust. But I would assume likely that they're using all that the skin flakes and anything else to grow. We were wondering, are they using anything from the carpet fibers themselves for growth? And when we imaged the carpet materials with the dust, when you actually get really down into it, they don't seem to be eroding or using the carpet. They kind of just grow around the fibers and on the dust.
Cool,
Yeah.
thanks.
Yeah. Oh, William asked a question, because I was just talking about SEM. Did you observe preferential microbial growth at near certain types of composition of dust particles? So with the SEM work, we didn't do any kind of characterization. It was more just what can we see. So we weren't able to say like, oh, in this image, we're seeing this specific species. We didn't identify at that level, but that would be very interesting if, there's certain dust particles that are there and it's causing this type of fungal growth. Yeah, that's a good question. Yeah, I don't know. I can keep scrolling through the chat if you want. I think there's one more. Where the spores part of the dust. Oh, I can go back to those pictures and we can look at because I think those pictures were really interesting. So they were asking about the dust. So these were the dust particles. So this is more zoomed out, the 50% image. And then the 95, I think we zoomed in a little bit more, and you can actually see these are the spores and the spore chain. So that's the actual fungal growth. And then perhaps something like this here, those would be the actual like dust particles that are sprinkled throughout. But this is the hyphae and the actual growth of microorganisms.
This was my question, actually. I think I meant more like you mentioned specifically that you need dust for spore growth to occur. But my confusion came from, if there is no dust, where does the spores come from? Like,
oh, sorry. So there's
spores be a subcategory of a type of dust?
Yeah. So the spores are from the microorganisms themselves. So that like the spores and the chains, like that's the actual microorganism that's growing. If there was no dust, so I don't have pictures of just carpet fibers without dust. That would have been interesting to do. But if there was no dust, then there was just no growth that occurred. So we wouldn't see any spores or spore chains if there was no dust in the carpet. It would just be carpet fibers. Does that make sense?
Yeah, that answers it. Thank you.
Great, yeah.
I think we're close to the hour. I had one final question, but it may not be a great one. So we often hear about mold formation when there's serious flood-in events, and that's one of the ones we're interested in. So, and you talked about the duration, how long it has to be at high humidity. So is it that if we have serious flood-in, everything gets wet, we have high humidity in a space until things dry out? Right. Is there sort of a time, if people can get things dried out within a day or two, they're okay, but if it's a week, they're in trouble? Or how do we think of that?
Yeah, so what I was seeing with, and specifically just with the carpet work, we were seeing that after 6 hours of elevated exposure, you could be seeing growth. So then that's really quick, right? That's super fast. So I think it's really hard to say, that it's this amount of time that if you had a severe flooding event. Ideally, my go-to is if you had a severe flood, if you had flooding and you had carpet or some kind of fabric materials, those are probably not going to be remediated to a full extent where there's not going to be any more growth, because it's very hard to get rid of water. But maybe that's a further down the line research of how long do we need, how long after a flooding event can you say this is okay? Because ours was really quick. We were seeing growth fairly quickly. And
I think for drywall as well, where
drywall as well. Yeah.
Longer, but everybody's got drywall. So
yes, true.
Yes.
Yeah. And what do you do? How do you how do you get it out of the drywall? How long does it have to dry out? Yeah.
Well, thank you for a fascinating and, as I said before, somewhat scary presentation.
I'm sorry.
It's good.
Just don't have homes with, just don't, keep it low. 50 50 to 60% relative humidity is okay.
Don't have carpet and vacuum and don't have flood. Yeah.
Yeah.
So there we go. So thank you for a fascinating presentation, and thank you for all who joined us today. So
Yeah. Thanks so much, everyone.
all right.
All right.
Thank you,
Mr.
Sarah. Bye.
Thanks.
Bye.
Bye.